Okay, everyone, welcome to the San Diego Chromatin Club. It's a pleasure today to introduce Dr. Oliver Rando. Dr. Rando was an undergraduate in Tom Maniatis' lab at Harvard, where he worked on the role of the proteasome in the activation of NF-kappa B. He then carried out an MD Ph.D. at Stanford in Jerry Crabtree's lab, working on chromatin remodeling and T-cell activation. He launched his independent career as a Bauer Fellow at Harvard, and then joined the University of Massachusetts Medical School in 2007, where he is now a full professor. Dr. Rando has made many seminal contributions to our understanding of chromatin structure using budding yeast as a model organism. His lab has developed novel methods to investigate nucleosome positioning, chromosome folding at single nucleosome resolution, and the dynamics of histone turnover and replacement. In related work, his lab was one of the first to map histone modifications at nucleosome resolution and perform functional genomics in mutant yeast lacking specific histone modifications. Ollie has also had a long-term interest in how epigenetic information transmitted through the germline can affect future generations. His lab has developed several mouse models in which they have shown that paternal diet or exposure affects metabolic properties in offspring mice. This is obviously of great interest as it demonstrates that prior environmental conditions could be a factor in metabolic disease risk today. His lab's current efforts are geared at understanding the epigenetic basis for this transgenerational effect. Ollie has received the NIH Director's Pioneer Award, Fellowship in the American Academy of Microbiology, and was a finalist for the Blavatnik Awards for Young Scientists. Welcome, Dr. Rando, and thanks for sharing your work with us.
Hello, thanks for that lovely introduction, and thank you guys for having me. It's good to see those of you I can see, and all of you I can't see. I wish I could. Well, so I'll go ahead and get started here. As Diana said, I am going to talk mostly about our work on paternal effects of environment today. So those of you who came hoping for yeast chromatin structure and function, I apologize, but we haven't really done anything interesting in that domain in at least like 5 years, so I don't really see what I would talk about. So my lab's general interest is in epigenetic inheritance, and so I'll start out just by defining the term because that term's been sort of polluted by a couple generations of people. So we prefer the 1970s definition of epigenetics from Holliday and Riggs, which is that a trait is epigenetically heritable if two organisms with the exact same DNA can differ in some phenotype, but they can then pass on that phenotype to their offspring. And so either through mitosis or meiosis. And so, you know, the famous example of this is cell state inheritance. To a first approximation, every cell in your body has the exact same genome. But when a liver cell divides, it never makes a skin cell or a kidney cell, despite sharing the book of instructions with those cells. You end up making two liver cells. And so this inheritance of state liverness is an epigenetic trait. Now, you know, decades of genetic analysis of cell memory systems has led to the identification of 4 or 5 molecular carriers of epigenetic information. This slide shows 2 of the 3 that we really focus on. So these include DNA modification, cytosine methylation in particular, and the packaging of eukaryotic genomes into chromatin, into nucleosomes. And of course, the Lewis trithorax and polycomb screens were really the foundational genetic connection between chromatin structure and cell memory. And so, you know, our work in yeast, a lot of our interest was in the question of how you replicate chromatin states, because of course, replicating DNA is quite well understood. But if that's like photocopying a sheet of paper, copying a chromatin state is like copying a piece of origami. So it's a much more interesting mechanistic question. What I'll tell you about today, though, is our efforts to understand epigenetic information between multicellular generations as opposed to mitotic epigenetics. So the main point to make here is that for multicellular organisms, this is not how we divide. In order for information to pass from one generation to the next, it has to go through the restrictive and disruptive processes of gametogenesis to form these 2 very unusual cells, the sperm and the egg. And then any epigenetic information present at the spark of life at fertilization has to then sort of over the course of development, have all the information needed to sort of set in motion a chain of events that lead to a physiologically coherent phenotype. So, of course, most epigenetic information in the germ cells is erased at fertilization. Otherwise, our children would be tumors made out of sperm or eggs. And of course, you can make all the cell types of the body. But it is the case that some epigenetic information escapes this erasure process. As far as I understand, the first discovered example of this was in plants. It was originally discovered by Bateson and Pellew in the 1920s in their studies of the rogue garden pea. It was then studied much more extensively in maize by Brinks and Co. in the 50s, who named the process paramutation. And so the basic idea is here. You have two different, the best studied case of paramutation. You have corn that's either green or purple. If you cross the green and the purple, you get green progeny. Now, if this were a Mendelian trait, this would tell you that green is big B, purple is little b, and these should be heterozygotes. Now, if you then crossed these heterozygotes to the little b's, you would expect half the offspring to be purple and half to be green. But that's not what happens. You end up getting all green progeny from this cross. And in fact, that will persist for hundreds of generations of backcrossing. And so this phenomenon was called paramutation because it sort of seemed like the inactive allele of this transcription factor was converting or sickening the active allele and converting it to the inactive state. And so over the last 40 to 50 years, people, especially like Vicki Chandler, have carried out genetic analysis of paramutation and found factors required for its propagation. And basically, she and others have found the three main epigenetic information carriers, chromatin structure, DNA modification, and small RNAs. All are sort of required for this transgenerational epigenetic inheritance system. So all of this, the discovery of molecular information carriers for epigenetic information, along with the fact that epigenetic information carriers are responsive to environmental conditions. So, if you heat shock a yeast, nucleosome modifications and positions change dramatically in the yeast genome. In contrast to DNA, which is largely unaffected by environmental conditions, this has led to the resurrection of an old idea, often called the inheritance of acquired characters or Lamarckian inheritance. It is obviously an idea that's had better days. Its best days are behind it. And so it's basically the idea that under some circumstances, it makes sense for parents to tell their kids about the world they experienced. Now, this was tested. I think the first serious test of this was carried out by August Weismann in the 19th century. So August Weismann is the German embryologist who defined the soma to germline distinction. And so what he did is he cut the tails off of mice and mated them to each other, measured tail length in offspring, and found no difference in tail length. And so this was an influential refutation of the idea of the inheritance of acquired characters. Now, there are at least two problems with that experiment. One of them is that maybe the useful thing to do if somebody's cutting your tail off, it's not to tell your kids to have a shorter tail, but it's to tell them to run away from people with lab coats. The other thing that even at the time, I think people should have realized is that Lamarck understood in developing his theory that amputees from wars throughout history don't have children without arms. And so even baked into Lamarck's theory of evolution was the idea that you would only tell your kids about the world if you wanted to, or if it was useful to you. And so even in the intellectual milieu of the time, this is sort of a little bit of a rigged experiment. Nonetheless, it certainly was quite influential for about a century. So this is the end of the history. The point of bringing this up is that, over the last 2 decades, we and many other groups have revisited this kind of paradigm. It's quite simple. You poke an animal and then you measure something in its kids. And so we've been doing this in the context of paternal effects in mouse. Okay, so briefly, what we do is we use inbred mice. So hopefully there's not a lot of genetic differences to be selected on in the germ cells. We split brothers to control and low protein diet. Low protein diet is what we've studied the most, although we've really sort of expanded quite a bit over the last couple of years into developing a bunch of other paradigms like nicotine exposure, high fat diet, stuff like that. We raise the males from weaning to sexual maturity. We then mate them with control females. We take the males out after a day, so the males certainly don't directly interact with the offspring. We also are trying to minimize things like microbiome transfer through the feces. Then the females are allowed to be pregnant, have their kids, and at 3 weeks of age, we grind the kids up to measure, instead of one quantitative trait tail length, we measure 30,000. And at the time we were doing this with microarrays to give you a sense for the era we're talking about. And so, you know, the differences between what we're doing here and what Weissman was doing, one is that I forgot to mention, we're doing this as a paternal effect because in mammals, mom is baby's first environment. So fetal alcohol syndrome is not magic and epigenetic, you're just poisoning a fetus. The other is that we are choosing an environment that it would make sense to tell your kids about. And I can talk about that in the questions if anyone's interested. And then finally, instead of measuring one quantitative trait, we measure 30,000. And so what we find, here's a sort of typical heat map for gene expression in the kids. I should mention we're looking at hepatic gene expression. Each of these columns is a comparison of one low protein offspring with one control. We labeled the low protein offspring's RNA in red and control in green. So these red patches are genes that are overexpressed in pups if their dad was eating a low protein diet. And so what you can see is that this is clearly not a sort of random puke of color, but we really get these fairly penetrant blocks of overexpression and underexpression of genes. It's not completely penetrant. We're probably at about 85 to 90% in this particular experiment. The green genes are not enriched for anything in particular, so the downregulated gene expression program doesn't really tell you much. The overexpressed gene expression program is very significantly enriched for lipid biosynthesis and cholesterol biosynthesis. We wanted to know whether or not this was just a gene expression phenotype or whether or not we could also get a physiological phenotype out of these guys. So by measuring a couple hundred metabolites in the livers, we find that the low protein pups have significantly reduced levels of free cholesterol and cholesterol esters. Okay, so this is as much as I'm going to say about phenotyping the pups, but people who know about metabolism tell me that a preferential effect on cholesterol esters suggests that we're looking at a bile acid phenotype. So perhaps what's going on with the pups is they're dumping a lot of bile acids to get more out of their food, and then they're homeostatically upregulating cholesterol biosynthesis to keep up with the loss of cholesterol. Okay, so that's our study. There are now hundreds and possibly thousands of papers documenting paternal effects of environments in mice. This set was all within a few years of ours. This paper actually is the one that pushed us out the door in 2010. These guys fed male rats a high-fat diet, and they document altered glucose control in the daughters. This is a system, this is basically our system carried out a couple of years later. And these guys found a bunch of other metabolic phenotypes altered in kids, including glucose control and things like blood pressure. I always point these out. These are quite interesting to me because in these systems, the investigators actually, it's in utero undernutrition. So what they do is they starve a pregnant female. They take her son, so he was in utero during that starvation, and use him as the paternal generation. And these are people who were carried out by people who know a lot more about epigenetics than I do, and understood that the establishment of imprinting, which is the best system for heritable epigenetic information in mammals, occurs during primordial germ cell development during fetal growth. And so these guys were trying to sort of more directly impact primordial germ cell establishment of epigenetic marks. Interestingly, they see pretty much all the same phenotypes that we see in weaning to sexual maturity perturbations. So glucose control, cholesterol metabolism, the rest of it. Okay, so these are all connections between a paternal nutritional status and offspring metabolism. These are reversible, so high-fat diet, the effects of high-fat diet on pups can be reversed by allowing dads to exercise. So that's another twist to this. Not only do paternal diets affect kids, paternal stressors, there's quite a bit of literature on things like early maternal separation or other kinds of stress to males affecting anxiety-related behaviors in their kids. And then the last big group of paternal effects paradigms are all based on various kinds of toxin exposure. The three I'm highlighting here are sort of nicotine, ethanol, and cocaine, so drugs of abuse, all of which affect various kind of motivational behaviors in their pups. And then there's a whole literature on things like endocrine disruptors from people like Michael Skinner and others. So in sum, there are, I would say, again, on the order of 1,000 papers now that have shown that the way you treat a male prior to mating can program health and disease in his kids. Now, these are all the rodent models. There's some evidence from other animals, some in pigs, sheep, and cows, and a little bit in humans for the same kind of thing. So the famous version of this, or the famous system for intergenerational effects in humans, comes from the very famous Dutch hunger winter. So this was a period at the end of World War II where the Nazis, in response to a transit workers' strike in the Netherlands, starved the Dutch people. So they, burned all the food and salted the fields. They were Nazis. Government rations got down to 700 calories a day. People were eating tulip bulbs to survive. Something like 20,000 people starved to death during this winter until the Allies restored food control in whatever it was, April of 1945. So what's interesting about this is it's rare for a first world country to go into a famine and come out with defined kinetics. And so people have found that children who are in utero during the Dutch hunger winter have significantly elevated rates of diabetes, obesity, and cardiovascular disease. Uh-oh. Oh, okay, my laptop froze up for a second, but it seems to be back.
Yeah, you're good.
Okay, sorry about that. All right, so, these are the same cardiovascular disease, obesity, and diabetes. These are glucose and lipid control phenotypes, a lot like we're seeing in paternal effect systems in mice. And actually, one point I always try to remember to make is that these are, of course, maternal systems, right? So these are children who are in the womb during the hunger winter, have these altered metabolic phenotypes. And one thing that's quite interesting is that all the phenotypes that we affect in paternal systems turn out also to be the phenotypes that are affected if in response to inadequate placentation. And I think that actually may be a really big hint for how these things work. There's a little bit of evidence for male line effects in humans. This largely comes from the so-called Uverkalix cohort, a remote parish in Sweden where they started keeping really good crop records in the 19th century for tax purposes. And so what Pembry, Bygren, Kati, and others have done in modern era is link your ancestors' food supply to morbidity and mortality. And what they find is that your paternal grandfather's access to food is correlated with obesity, diabetes, and the rest of it. And so this is not a particularly well-loved study. It doesn't have very high numbers, but it's some of the evidence in humans that, male line effects of nutrition affect metabolism in the future. So certainly in mice, I think it's more or less irrefutable at this point that a father's dietary lifestyle affects metabolic traits in kids. There's some evidence for this in humans. And so, the bulk of my lab now is very keen to try to understand how this works mechanistically. So here's sort of a cartoon just pointing out what we do. We take males under different conditions, mate them to control females, document altered stuff in the kids. And so naturally, the assumption / hypothesis is that this is going to be mediated by epigenetic information in the sperm, whether that be where histones are retained versus getting replaced with protamines, DNA modification patterns, or small RNAs. It's worth pointing out there are other ways dads can talk to kids. There's a great literature, largely in flies, but not only in flies, for seminal fluid peptides and small molecules, altering female post-copulatory feeding and mating. There's a really groovy literature in birds where females judge mate quality and change how they allocate resources to their kids. And so in principle, and then of course there's the microbiome, which isn't even on the slide. And so in principle, we could have our control females judging the low protein male and restricting blood flow to the placenta. So the first question is, where is our transmissible information? To get at that, what we do, and pretty much all reproduction in our lab has now converted to in vitro fertilization. So basically you purify germ cells from males under different conditions. So the sperm are purified away from the seminal fluid. You fertilize a control oocyte in vitro, and then you implant that zygote or 2-cell embryo into a second female, the recipient. And so we've been able to recapitulate our phenotype through IVF, which tells us that the information is in sperm or co-purifies with sperm. The other really useful thing about having become reproductive biologists is that we can now take maternal effects seriously, because now we're splitting the female side of the equation into the oocyte donor and the uterus donor. And so this is something that we're sort of actively growing in our lab right now. Okay, so we know the information's in sperm, as one would have expected. We are quite sure the answer isn't cytosine methylation in our system, which is what everyone in mammals kind of expected it to be. We're less certain that it's not chromatin packaging, but we don't even really know where histones are in sperm. Quite a few labs believe they're one place. We disagree with them, but it's not clear even if histones are retained penetrantly from sperm to sperm to sperm. So we're, let's just say, pessimistic about chromatin as a relevant carrier. And so we have really focused in the last 5 to 10 years on small RNAs in sperm. Small RNAs, of course, are the transmissible epigenetic signal in the really good transgenerational epigenetic inheritance paradigms: paramutation in maize, RNA interference in worms. So small RNAs are how parents talk to kids. So we spent a lot of time cloning small RNAs out of sperm. Typical cloning methods, we and about 50 other groups all get the same result. This is the insert size for the small RNAs in mouse sperm. This peak at 21 nucleotides is the microRNAs. That's most of what you'd get out of a fibroblast or hepatocyte or something. But of course, the elephant in the room is this dominating peak that's about 80 to 90% of small RNAs. I'm excluding rRNA mapping reads. All of which derive, they're 28 to 35 nucleotides, and they all derive from the 5' ends of mature tRNAs. So these are called tRNA fragments or TERFs. It's worth pointing out that in the last 2 years, well, in the last 5 years, we've recognized that typical cloning protocols were failing us. We actually know that the 3' ends are there, but just unclonable. tRNAs turn out to be really hard to clone because they have a bunch of nucleotide modifications that block RT. And so we really have to revise our view of what sperm are carrying. Actually, I think every epigenome measurement in sperm is a little bit wrong, at least. But in any case, I'm just going to tell the story focused on the 5' ends. But it's worth noting that we can now clone the 3' ends, and they're also very abundant. And that changes how we think about stuff a bit. Okay, so we've been chasing, so what we find in low protein sperm is upregulation or, overabundance of this tRNA fragment, TERF glycine GCC. It is the third most abundant RNA in sperm after GLU, CTC, and VALCAC. It's about 5% of all small RNAs that you can clone the normal way. And we sort of got into this, we decided to chase this down for a bunch of reasons. First, of course, you would tell your kids about a low protein diet using a small RNA made from tRNAs. Small RNAs are how you talk to kids. tRNAs see metabolism in a very obvious way. This is very abundant. And so for sperm to do anything meaningful to the zygote, it's going to have to be a really abundant RNA that's affected because, of course, the oocyte is 1,000 times bigger than sperm. And the last, which I'm not really going to get into, but this was actually what motivated us to chase tRNA fragments. tRNAs are universally used in LTR element replication, both things like HIV and exogenous retrovirus, but also endogenous retroelements, transposons. So tRNAs are in the wild, they're the primers for reverse transcriptase. And so you can imagine that by cutting your tRNAs, you could fight off an LTR element. So, people talk about piRNAs as a genomic adaptive immune system. tRNA fragments could act as a genomic innate immune system. Any LTR element I could get from another human being, I could fight off by clipping my own tRNAs, even if my genome's never seen it before. And because epigenetics is about control of selfish elements first, that then gets co-opted for other forms of regulation, this got us really keen on these tRNA fragments. Okay, what's our next slide? All right, so at this point, our lab is kind of interested in every aspect of this pathway from how dads sense the world. So what are the molecular changes in blood that affect cell type X, Y, or Z that then affect the molecular contents of sperm? What are the changes in the sperm epigenome, which I just talked about? And then how do molecular changes in sperm, which don't persist very long in the zygote, particularly small RNAs, there's no copying mechanism for small RNAs in mammals, how do those affect gene regulation and set in motion the chain of events that lead to altered glucose control, say? So I'll start up here, which is that, naturally the dogma about small RNAs in sperm is that or all RNAs in sperm is that they're the degraded remnants of the RNAs you use to make the sperm. And so the null hypothesis would be that our low protein testes have higher levels of tRNA glycine GCC. And in the course of chasing that down, we sort of became convinced that the small RNAs that sperm carry, at least in part, if not completely, were not made during testicular spermatogenesis. And so this now means we have to learn about the male reproductive tract. So in mammals, sperm develop in the testis. At the completion of this process, you have a morphologically mature sperm, which exits the testis but is incapable of fertilization. They don't know how to swim, among other things, and they're lacking some protein receptors for the oocyte. So sperm then spend, depending on the mammal, between a week and 2 weeks transiting a long convoluted tube called the epididymis. They go from caput to corpus to cauda. The cauda is the storage organ for ejaculation. So this is where, if you do IVF with mouse, this is where you get your sperm from. And then ejaculation is basically a muscle contraction around the cauda that squeezes the sperm out the vas deferens. The epididymis is the least studied organ in the body, in part because it apparently doesn't get cancer, which is kind of interesting. It was described in the 17th century by the Dutch anatomist Reinier de Graaf. So you can see here how you fit 2 meters of tube into this little nugget of tissue that envelops the testis. My favorite factlet about the epididymis is that in a 2005 German newspaper poll, it was ranked people's 3rd favorite male reproductive organ, which, A, means we beat the prostate, but B, is sort of a ridiculous poll to have carried out. Anyway, so the epididymis is, really a not particularly well understood organ. Here's a light sheet image we made where you can see the tube, about a meter of tube all coiled up. The epididymis is broken into these segments, 10 in the mouse. And all of them have very different gene expression profiles, and they create very different luminal chemical environments and protein environments in the lumen. So sperm are basically spending 2 weeks incubating for a day or two in each of a bunch of different, soaking baths. And so what we think we found is that this tissue is responsible for synthesizing the RNAs to be delivered to sperm that will be delivered to the zygote in order to alter offspring phenotypes. So yeah, I have a little evidence for that here. So in the testis, you don't see any tRNA fragments when you clone small RNAs, and that's true both for the bulk testis, but also if you purify various germ cell populations. The green in these pie charts is piRNAs. Here's the data I showed you earlier that cauda sperm are filled with tRNA fragments, and we find earlier in the epididymis that caput sperm are also filled with tRNA fragments. So the last sperm we can get from the testis are filled with piRNAs, and then they are epigenetically remodeled to switch from piRNAs to tRNA fragments. I will point out there are a number of about 100 RNAs that change abundance through the epididymis as well. We find that, here's northern blots for tRNAs. There's no tRNA cleavage in the testis, as I told you. In the epididymis, in the epithelium, we see very robust tRNA cleavage. So sperm exit the testis without tRNA fragments. They enter a tube where the epithelium is making tRNA fragments, and during this entry, they gain the tRNA fragments. What we know about, one of the things we know about epididymal transit is that the epididymis ships proteins required for fertilization to sperm in little vesicles. It's a combination of exosomes and microvesicles, collectively known as epididymisomes. Now, exosomes are very famous for carrying RNA from one cell type to another. And so we clone small RNAs out of these epididymisomes. We call this epidemiological epididymisomal epigenomics, or as Job Decker always wants me to call it, 3E, which I think is cute. And so sure enough, epididymisomes have the exact same small RNA payload that sperm will gain. So here's a scatter plot of cauda sperm versus epididymisomes. The green under the diagonal here, these are piRNAs, the exception that proves the rule. So those are present in sperm, but not epididymisomes because they were carried forward from the testis. But everything else more or less falls on this line. Sperm and epididymisomes are better correlated than any of the small RNA populations, testis versus epididymis, sperm versus testis, et cetera. Finally, we can reconstitute this delivery ex vivo by fusing testicular sperm with caput epididymisomes and repurifying the reconstituted sperm. This green line are the piRNAs, which are unaffected and tell you something about the technical reproducibility of the assay here. Everything above this line was delivered by this fusion process. So we're pretty sure, although I have to say I've never been able to get over 95% sure of this, but we're pretty sure that the epididymis is synthesizing the RNAs that sperm are going to deliver to baby. Okay, so whether or not the epididymis, so this positions the epididymis as a novel sensory organ for environmental conditions to program sperm. And it sort of makes the more general point that there are a number of cell non-autonomous ways for RNAs from other cell types to make their way into sperm. What people would have assumed, and we have actually a little evidence for this now in a different system, people would have assumed that Sertoli cells would play this role. Of course, I told you about the epididymis and the prostate are both well known to generate lots of vesicles carrying RNAs. And then there's evidence in some other organisms, worms in particular, for definitive non-reproductive tissues like the brain to ship RNAs to the germline. So I'd now like to talk a little bit about our efforts to understand how Dad senses the world. So insofar as we think the epididymis is sensing the environment and modulating sperm RNAs, the assumption would be that the epididymis gets a blood supply, and so circulating leptin levels or insulin or LDL or something are received by the epididymal epithelium and alter RNA production somehow. a postdoc in my lab, Vera Rinaldi, came to my lab with a very, very different idea based on an experiment she did just before she left John Chimenti's lab. So she had a friend who was doing connectomics in the brain. So they use this reporter mouse called AI14D. It's basically a Cree-driven GFP. Specifically, it's a promoter, locks, stop, locks, tomato. In the absence of Cree, the tissue's black because you're just driving the stop cassette. In the presence of Cree, you get red cells. So what this guy was doing, Andrew, was injecting the prefrontal cortex of these mice with a lentivirus or AAV anyway. Let's forget if AAV is a lentivirus. Carrying Cree and then mapping tracts in the brain. You get these beautiful images. They're just unbelievable. And so Vera said, "Hey, that's great. You don't need the rest of the carcass, do you? Can I get a hold of it?" And so she looked in the liver and the lungs. The liver, liver, lungs, and kidney. And doesn't see any fluorescence. So this is where, if Andrew had been like missing the brain and hitting the jugular, this is where you would worry about AAV infecting cells. She didn't see any fluorescence in the testis, which is what she was looking for. But remarkably, she ends up finding really robust tomato expression in the epididymis. And these are really beautiful images, and we have a bunch of them. First time she showed me this, she said, the second brain isn't where we thought it was. But so you can just really see the cauda epididymis and the vas deferens lighting up. So that suggests the possibility that molecular stuff made in the brain is making its way to the male reproductive tract. So, with an AAV experiment, you worry that it's just a weird tropism. And so she then turned to double transgenics. So here she's just crossing the reporter animal with various neuronal Cree drivers. This is FEV. It's one of the ones we've used, which drives serotonergic neurons. And she finds in these double transgenics, we get a red epididymis. So these two experiments together it's very hard to see how they are both artifacts. With the AAV experiment, you worry about maybe AAV has an unexplained tropism to the epididymis because no one studies the epididymis. But of course, that can't explain a double transgenic. In the double transgenic, Cree drivers are famously leaky, particularly in the male reproductive tract. And so, so you always worry about that kind of thing, but that wouldn't explain the AAV experiment. So it really seems like Cree information synthesized in the brain is making its way to the male reproductive tract, which suggests a very different way of viewing how environments are sensed, which is that the central nervous system plays a role in interpreting environmental conditions, extracting salient features and, compressing them somehow, and telling the epididymis what to tell sperm. We're doing not too bad. Okay. This is, why am I showing you this? A couple different neuronal subpopulations have light up different parts of the epididymis. So here's chat, this is cholinergic neurons. This is actually where this image was pulled from. And here you can see the initial segment of the epididymis lights up like crazy. And this has the exciting implication of maybe different emotional or motivational states, each sort of tune different parts of the epididymis. So, if you're in a serotonin-y mood, maybe you squeeze segment 8 of the epididymis and that gives you, more Valcac or something. Let's see, this is just regular histology to show you what it looks like in the epididymis. So the cells that are receiving Cree are the principal cells of the epithelium. They are not the basal cells or the clear cells. You can see this very patchy reception, which is kind of interesting. We're trying to figure out what distinguishes the recipient cells from others. The other thing you should notice here is that, here's nestin Cree, which is a pan-neuronal Cree. This is adipinectin. So when we started this, we thought this was a one-to-one mapping from brain to male reproductive tract. Now as we've delved into this further, we find other Cree drivers like fat tissue will also light up the epididymis. So the epididymis is really receiving Cree from all over the body. Okay, we've been trying, so the last thing I'll say about this is that we've been trying for years to definitively test the idea that the Cre information is coming through the circulation to the male reproductive tract. And we've done that two ways. One is with parabioses, where you stitch together the circulations of two animals. It's used in aging research a lot. So you stitch together a reporter animal and a neuronal Cre and see whether or not you get recombination on the reporter side. And then the other thing we've done is taken either serum or circulating exosomes from neuronal Cre drivers and injected those exosomes into a recipient. Here's sort of a negative control. I think it was injected with exosomes from an FV, just a wild type animal. And here's the transfer from one of the neuronal Cre drivers. I can make this look really great because I can show you the one experiment where it worked. But the problem is that we've been doing this for a couple of years now, and we've done something like, 5 parabiotic pairs and 40 or 50 serum transfers, and maybe 3 animals show recombination. So on the one hand, I can't explain how this would occur if there weren't circulating Cre that made its way to the epididymis, but it really is not particularly reproducible. And it's so inefficient that it's going to make it very hard to do any of the next stuff one might want to do, like fractionate where the activity is in the serum, or ask whether or not there are functional consequences for epididymal physiology. So anyway, that's sort of where we are now. Even though I can't really close the story here, it is pretty much the most exciting thing that's happened in the lab in the last couple of years. And so I just want to leave everyone with the idea that, the way that the germline sees the world, you really shouldn't just be thinking about the germline proper, spermatogonial stem cells and on, but there are a wide number of tissues, dedicated reproductive tissues and non, that may play roles in how dad sees the world and in programming kids. We're trying to get at this more systematically using ligand receptor interactions. So we actually started with nicotine, which binds to nicotinic acetylcholine receptors. That turned out not to require nicotinic acetylcholine receptors. More recently, we've been using other ligands like rosiglitazone, which is a PPAR gamma agonist. So the idea is to, hit males with rosiglitazone or nicotine or whatever, identify some change in epididymal small RNA production or offspring phenotype, then use Cre-Lox genetics to specifically delete PPAR gamma in this case in the epididymis or Sertoli cells or the brain, and then see which of these Cre drivers will prevent sensing and modulation of kids. So this is something that I think we're really, I think has to be the future of the paternal effects literature. Okay, so that's how dad sees the world. I have about 15 minutes left. I'm supposed to stop at 1, or am I supposed to stop 5 to 10 minutes early?
I think, yeah, just maybe 5 minutes early for questions.
Okay, great. So downstream of changes in the sperm epigenome, we've spent quite a bit of time and effort trying to understand how, so this is now moving from here down to here. We're really trying to understand how small RNA changes in sperm or, other things like cytosine methylation changes alter preimplantation gene regulation and development. We do this, using commoditized single cell techniques, single cell RNA-seq or cut and tag or whole genome bisulfite sequencing. So, the idea is basically we can either take sperm from males who were treated different ways, or we can inject small RNAs or siRNAs against a chromatin remodeler. And then we can sort of sequence all the RNA in throughout preimplantation development. We use SMART-seq2, which was developed for single somatic cells like fibroblasts. There you capture something like 3,000 to 5,000 molecules. What's nice about the early mammalian embryo, it's a little bit of a hassle, you can't make a lot, but they're so large that we've now used unique molecular identifiers in some of our sequencing, and it's clear that we're capturing over 20 million molecules. So actually one zygote or 2-cell embryo has a quality of RNA-seq data that looks like, you've ground up a whole liver or something. So this was our first, whatever it was, 400 embryos. And you can see by PCA, we really nicely separate early development. Principal component one is the degradation of maternal transcript. So that proceeds, sort of monotonically from the oocyte to the blast. Principal component two is zygotic genome activation, which in mouse happens at the middle 2-cell stage. So you turn on the zygotic genome, carry those RNA products, and then they are slowly degraded. So it turns time into an angle. So, let's see. So these are all the different ways that we perturb RNA levels in zygotes. We can either, the obvious one is to like inject our favorite tRNA fragment into a control embryo. We can use Krelok's genetics to eliminate things like microRNAs in sperm and ask how that affects things. The idea that I sort of like the best, it's sort of the cutest, and it relies on our epididymal work, is to use sperm from different parts of the male reproductive tract, the testis where you don't have any tRNA fragments, or the caput or the cauda, which differ in the abundance of various microRNAs. Carry out ICSI. So, these sperm don't swim, but because of human-assisted reproduction, we as a community have the ability to make embryos by just injecting the sperm into the oocyte. And then we can ask what the difference is between fertilizing an embryo with a heavy TERF versus a heavy piRNA payload. And then last, and probably the most generic, is to basically make embryos that don't have any sperm contribution at all. So these are parthenotes. You activate them with strontium chloride and cytoclasin B. They're really messed up, of course, because all of the imprinted genes are misregulated, but they proceed to blasts just fine. And so we've been doing a lot of work just taking total sperm RNA and injecting it into parthenotes, because parthenotes don't have any sperm contributions at all. So it really gives us a blank slate. Very briefly, I'll touch on the best developed of these stories, which is using sperm from different parts of the epididymis. So this was Colin Conine. He took caput sperm and cauda sperm, made ICSI embryos, and carried out single embryo RNA-seq. We have identical fertilization rates and blast progression rates. So the caput sperm are not grossly aneuploid or oxidatively damaged or something. And what Colin finds is overexpression of about 50 genes. They're all RNA and chromatin regulators, which is a very interesting, really interesting profile. And it's really reproducible. So here you see it at the 4-cell, 8-cell morula blast. And in fact, it's so reproducible that we were able to use this to fractionate the activity responsible for this. So basically what Colin would do is make a caput ICSI embryo. The eggshell hardens up and then softens again after 3 hours. So we wait 3 hours to inject RNAs. And then we ask, what RNAs are responsible for the overexpression of these genes in the caput embryos. So here's one of our favorite targets, HNRP-AB. So here are cauda-derived embryos in caput. You see we're overexpressing this gene. If we take gel-purified small RNAs from cauda epididymosomes, you completely rescue this. When we separate the small RNAs by size into TERFs and microRNAs, the TERFs don't do anything in this context. It's not totally surprising because caput sperm have almost all the same TERFs as cauda. But microRNAs actually that are missing from caput sperm will suppress this program. We've sequenced small RNAs out of sperm. We know which ones are missing from the caput. We can synthesize those and they will rescue as a group. This tells us that the microRNAs don't need to be specially covalently modified or preloaded into an effector like an Argonaut. And then the rescue is less good here, but it really seems like it's a really interesting microRNA cluster called the MIR880 cluster on the X chromosome. And you can see here, it's very weird. It's present at high levels in the testis and high levels in the cauda, but it's 100-fold less abundant, all of these entities, in caput sperm. So anyway, Colin just started his lab at Penn, so he's going to sort of chase down what MIR880 is doing. I'll also point out that this particular paper has been really controversial, which is crazy because it's probably the best documented result we have. But Colin wasn't able to make offspring out of his caput embryos, and everyone else in reproduction can, and it made people very angry. Fortunately, we knew that when we published the paper, so we spent a page talking about the technical differences between what we do and what the previous group had done. And it turns out that the difference between, the answer turned out to be the last one I would expect, but it's actually how you make your sperm head. So if you sonicate sperm to break the heads and tails apart and make caput embryos, those make pups. That's how people usually do it. If you break the sperm through fine needle shearing, like we do, which we thought was going to be less of a perturbation, you can't get pups out of caput sperm. So that's another thing Colin will have to figure out, is what's the difference between these two ways of making sperm heads. All right, I've got maybe 4 or 5 minutes. Okay, so at the very end, I'd just like to point out our focus on our favorite diet-regulated tRNA fragment, turf glycine GCC. And so, tRNA fragments are the least studied class of small RNAs. For a long time, they were assumed just to be degradation products of abundant stuff. They're increasingly being taken seriously as regulatory molecules in their own right. And so when we mess with turf glycine GCC, either transfecting it or knocking it down in ES cells, we get a very characteristic outcome. This is just mRNA abundance by, I think this was Affymetrix microarrays. When we knock down our turf, we see de-repression of 75 genes or so. It's very specific to knocking down the turf. It's not knocking down the intact tRNA. All of these genes are really well known to mouse embryologists. They all use as a regulatory element the long terminal repeat of MERV-L, an endogenous retroelement. So this was really exciting. There's this ancient link between tRNAs and ERVs, but actually it's a total red herring, because the way turf glycine regulates MERV-L has nothing to do with homology to the primer binding sequence. And we knew that instantly because, MERV-L, the L stands for I use leucine tRNA as a primer. And many of these genes are regulated by solo LTRs, where you've recombined out the meat of the ERV, including the primer binding sequence. So these genes don't have the primer binding sequence. These genes are also very famous because they are responsible for the zygotic genome activation program. So in mice, when you wake up the embryo, you turn on MERV-L itself and about 300 genes that use MERV-L as their regulatory element. That's off by the time you're a blast. And there's this beautiful work from Pfaff and McFarlane who showed that not only is MERV-L on when cells are totipotent, but actually MERV-L positive cells are functionally totipotent. So a colony of ES cells, all the cells are OCT4 positive, most of them, I mean. If you take any of these cells and aggregate it into an embryo, it contributes to inner cell mass, but not to trophectoderm, and therefore to embryo, but not to placenta. This is why ES cells are pluripotent, not totipotent. When they sort out the MERV-L positive cells from an ES cell culture, about 2% of an ES cell culture cycles into this MERV-L positive state, the 2C-like state. These, when aggregated into an embryo, contribute to trophectoderm and to inner cell mass, and so to embryo proper and to placenta. And so I mentioned that all the phenotypes you see in paternal effects are also affected by altered placentation. And so our working hypothesis is that by either affecting the amplitude or the perdurance of the totipotent state, turf glycine GCC is affecting lineage allocation into inner cell mass versus trophectoderm, and therefore, investment in placental function versus embryo growth per se. So we think that paternal effects are all basically very, very cryptic maternal effects, where dad is programming the placenta. We've worked out how this tRNA fragment works, but I'm out of time. But basically, ERV elements are opportunistic about underassembled chromatin. And it turns out that our favorite tRNA fragment ends up regulating through the unique histone 3' UTRs, the production of histones. And so here's the evidence for it. It's in Genes in Development last year, so I'll just mention the paper. So this is kind of how we're thinking about things now, is that by altering histone production in the preimplantation embryo, our favorite tRNA fragment is affecting how quickly you wake up the genome, and therefore affecting placentation versus embryo growth. Anna has now gone ahead and injected zygotes with turf glycine alone, which I always thought was not a great experiment because I thought some of the other misregulated RNAs probably also played a role. But she actually gets massive pups out of these guys when you inject our favorite turf. There's a really nice element of specificity because when she adds the 3' turf to the 5' turf, it inhibits the large or suppresses the large pup phenotype. And so our favorite RNA actually does induce quite a large phenotypic effect in the next generation. Okay, the last idea I want to leave you with then is, waking up the embryonic genome early versus late really doesn't tell you anything about low protein diet in particular. It doesn't seem like a specific signal. If dad wakes you up early, it could be Christmas or your house could be on fire. And so this plugs into a longstanding interest of ours, which is how much information do dads tell kids? With cytosine methylation, you have 25 million CpGs. Each of these could be a bit. And therefore you could tell your kids, nearly infinite amount of information about the world, cadmium levels in the water, average temperature, where you could find mates or fruit or whatever. And so here you could have a very high bandwidth view of the world that you tell kids. Alternatively, there's lots of reasons that to think that you just tell your kids about overall quality of life. Life was shitty, okay, or great. This is as good as it gets. And so for a bunch of reasons, we sort of favor these low bandwidth types of hypothesis. You can imagine something that's sort of intermediate, right? So maybe GLU-CTC tells you about psychic stress, GLI-GCC tells you about nutritional status. But anyway, this I think is one of the deep questions. If you allow that dads tell their kids something, then the deep question is how much? Well, not the deep question, the obvious question. Okay. Anyway, so here's just the cartoon to fix in your mind some of the stuff I told you. Hopefully I've convinced you that a dad's environment affects health and disease in his kids. I hope you have gained a new appreciation for the epididymis, which is a really fascinating tissue. And for the general idea that non-germ cells play some role in programming germ cells. And then we're very keen to understand how an early change to histone levels end up causing huge difference, significant differences in offspring size and metabolism. Okay, so the people to tell you about here, Upasana Sharma really did all of the epididymis work early in our lab. She sort of made our lab an epididymis lab. She just started at UC Santa Cruz a couple of years ago. Colin Conine did the ICSI stuff I told you about. Anna Boscovich and Xinyoung Bing worked out the molecular biology of our favorite tRNA fragment. And then here's a modern view of our lab from last May or something. And I just want to highlight here Vera Rinaldi, who just left abruptly, which was a bummer. But she was the one who did all of the brain to male reproductive tract stuff. And with that, I would gladly take any questions. Thanks for your time.
Great. Thanks so much. That was super thought-provoking. And we already have a bunch of questions. Everyone just go ahead and use the Q&A feature. But I'll start it off since we've already got a list here. Okay, so Udistar Betty has a question. Based on environmental sensing by the epididymis, do short exposures, i.e. 12 to 14 days in mice, recapitulate the effects of chronic low protein diets?
Yeah, so we've never done kinetic stuff. We've never done half weaning to sexual maturity halfway on low protein diet than control or other kinds of switch. In the last year, there have been several papers that have documented paternal effects induced by stimuli a week before mating. So Brett Nixon did this with acrylamide. I believe Isabel Nansui and Eric Miska have done one with stress. I know Tracy Bale used to tell me, I don't know if she's published, a week-long, stressing males and then a week later mating them. Those are all very gratifying because while I'm very suspicious of the epididymis still, the fact of the matter is it takes 10 plus days for sperm to get from the testis to an ejaculate. And so for a stimulus a week before mating to affect the kids, it cannot be affecting the testicular process of gametogenesis. So that's about as much as I have to say about that. But so there are some very short-term stimuli that have been documented to affect kids.
Okay. I think a follow-up, but maybe similar response is, do sire mice put back on a normal diet produce normal or improved offspring within the same window?
Yeah, so we also haven't done that. I know Jamie Hackett, he's given a talk about it, so I suppose I can share it, even though it's not published. So he's studying paternal effects of eliminating the microbiome. So it gives non-absorbable antibiotics. And their system is really beautiful. So they get a pup size phenotype. But then when they remove the antibiotic, the microbiome recovers.
Mhm.
And after it's fully recovered, they start producing pups of normal size. The only thing that we've contributed to there is that we have helped Lori Goodyear with a paper where they had males on high-fat diet and then either exercised them or not. And the exercise can reverse the offspring gluconeogenesis phenotypes. So, all of this is, so it does suggest that, you make a mistake early in your life and it doesn't permanently mess with your sperm.
Right.
On the other hand, it has the less sensical implication that, like, you tell your kids what just happened to you. Like, I had some 7-layer dip before I came up to give my talk. And if I, had a kid right now, would that kid learn that the world is filled with 7-layer dip, right? You would think that there'd be some, like, low pass filter where you need to experience extended periods of something for it to be useful to tell your kid. So that's kind of surprising. But anyway, that's the data. A bunch of people have done short-term exposures and gotten their kids programmed.
Right. Okay. From Merrick Rumke, great talk. How long is brain information retained in the epididymis?
Yeah, so the way that system works is we're using Cree locks, and so that has basically infinite gain, right? If you have recombination induced by a single molecule of Cree, you're just as red as a cell that had recombination induced by 1 billion Cree molecules, and so that's probably the most troubling aspect of that data, because we want to be able. There are two ends of the spectrum of what that data set means. One is the interesting one, which is, the brain makes information to program epididymal physiology. The other is that the epididymis is a trash can, like it, just circulating material gets through epididymal epithelium at some frequency. And so to test those two, what you'd want to do is ask, what things that don't have the gain of Cree due to the epididymis. So in other words, stress an animal, measure something that changes in the epididymis, then stress an animal and ship his blood to a second animal and see if you've altered the epididymis.
Mm-hmm.
And that has turned out to be technically impossible because the transfer experiments are wildly inefficient.
Right.
So basically, I can't answer that question currently, and I don't see a productive way, a path forward to being able to answer it without dedicating like a 10-person lab full-time to it for years. So
right.
I may never know the answer to that question.
Okay. All right. M. Donahoe says, what cleaves the tRNAs in caput and cauda? Is there a particular cellular compartment in the segments of the epididymis that the TB tomato transgene home to?
So the first question, we thought we knew the answer to that. So the famous tRNA nucleases are RNase A and T family members, and L. And we're pretty sure that's what's causing the cleavage, because we know the 3' ends of those tRNA fragments have cyclic 2', 3' phosphates, which are left by those kinds of nucleases. And so there's an RNase A paralog in mouse, or a family of them, called RNase 9 through 12. They're totally epididymis specific, and they are massively expressed in the epididymis. So RNase 10 is like, in boars, it's 70% of the luminal protein in the caput epididymis. It's huge amounts. So Upasana started her lab in part to delete those and eliminate tRNA cleavage. And the sad trombone music is that that deletion actually doesn't seem to affect tRNA cleavage. So I guess the answer is we don't know who the nucleus is. It's probably RNase T2A, but we don't know. As far as the compartment question, the Cree is flipping principal cells largely in the cauda and vas for most neuronal Cree drivers. But we do see some recombination in the caput and corpus. And then this one Cree, the chat, is really very initial segment specific. But largely it's the cauda that's receiving this Cree communication.
But just to I guess clarify, you believe that the Cree protein itself is being transferred rather than TD tomato.
Yeah, and we're let's see, we're sure of that. Well, we would be sure of that in the sense that if we believe the transfers from the Cree animals to the reporter animals. Of course, the Cree animals don't have the reporter cassette, so it would have to be Cree activity that was being transferred.
Mm-hmm.
The other, we know that it's Cree being transferred because when we assay recombination on genomic DNA from the epididymis, it's recombined.
Right. Okay.
So it's not tomato, it's Cree.
It's not tomato. Yeah. Okay.
We think it's Cree. We suspect it's RNA. We can detect RNA in the blood, or in exosomes. We haven't detected protein, so we think it's Cree mRNA in exosomes, but we don't really know that.
I see. Okay. Okay. Adam Cabede says, thank you for the amazing talk. I was wondering, is there a pre-fertilization time window during which environmental insults on dad could be passed on to son/daughter? In other words, how much of this small RNA information is retained over the lifetime of dad, or is what matters what dad did in the hours, days, weeks immediately preceding mating? So
yeah, so that's very similar to the first question.
Yeah.
I mean, again, we haven't done that kind of stuff, but we know that high-fat diet is reversible by exercise, and we know that multiple labs have done, roughly a week to 10-day stimuli and got pups affected. The most recent would be a Cell Reports paper from Natalie Trigg and Brett Nixon on acrylamide exposure. So they were very careful about the timing of exposure. So if you're interested in that question, I suggest looking up Trigg and Nixon 2021.
Okay. Jason Lynn, really cool work. I'm interested in the persistence of the signal in sperm small RNAs. How long does it take for the father to clear out the environmental signal and to return to normal?
I think the same answer to that question as well.
Paul Soloway, does control male sperm exposed to epididymal exosomes from low protein fed males produce offspring with metabolic perturbations induced by low protein diets?
Yeah, that's one of the classic questions I get. So we've never done that. One thing you probably didn't notice because I was going fast, but when we reconstitute testicular spermatozoa with caput epididymisomes, after 2 to 4 hours of fusion, which is about as long as sperm will hang out in vitro without dying, we delivered RNAs, but we did not convert them completely to a TERF-dominated profile. So normally sperm spend 2 weeks, and we just can't get that delivery of that order of magnitude to occur in vitro. So we haven't even tried the experiment because I just think it's kind of hopeless. But there are at least 2 and possibly 3 labs who have done that experiment and report that it works. Probably the best of them is from Humanix and Rompola, who study paternal ethanol. They fused sperm with ethanol-exposed epididymisomes and got some of their phenotypes out of those sperm-fertilized pups. Tracy Bale has done it. She did it a little bit more weirdly. She took an epididymal cell line and put cortisol on them, then took their exosomes, fused them with sperm, and made pups. So, and then I think Elizabeth Mansui's done it as well, although she might only have done measuring RNA changes in epididymisomes. So anyway, at least the Bale and Humanix papers suggest that experiment would work. I'm really surprised because I just don't think you delivered that much RNA ex vivo because you just don't have that much time.
Right. Okay. Okay. Anonymous says, egg cells have their own TERFs, question mark, and how does sperm TERFs regulate metabolic genes expression in the liver of the offspring?
Oocytes do have some TERFs. It's a much lower fraction of the small RNA payload of oocytes, which are sort of dominated by endo-siRNAs and microRNAs. But because the oocyte has so much more stuff, it's kind of an open question of how much, even if oocytes are 10% TERFs and sperm are 90% TERFs, and oocytes 1,000 times bigger. So, one of the damning questions that we'll have to address at some point is, how is it that sperm contributing like another 1% of a given TERF affects the zygote? So I don't know the answer to that question. And it's a serious question, and it's a serious problem. In terms of how our favorite TERF makes the big pups, again, we think it's very indirect, right? So mammals don't have a way of copying small RNAs, like worms and plants have RNA-dependent RNA polymerase.
Mm-hmm.
They can maintain high levels of small RNAs. Mammals can't. And so the small RNA really has to act within the first cleavage or two. So, our TERF does regulate genes expressed at the right place at the right time in the two-cell stage. How, affecting histone levels / the ZGA program in the two-cell embryo turns into a metabolic phenotype, again, our speculation is that it's by somehow affecting placental function or size.
Mm-hmm.
But we really haven't done any serious work to address that. It is what the lab's moving towards, but we have not tested that hypothesis, but that is our hypothesis. It's via control of the placenta.
Okay. I'm just confirming you're okay to hang on for another 8 questions.
Absolutely.
Okay, sorry. Okay, from Anonymous. Abundance of the selected small RNAs appears to be heterogeneous among the sperm cells from both control and treated male mice, with a large overlap in their sperms between control and treated mice. If these RNAs are essential in meeting the intergenerational inheritance, do you expect that only the sperms with the most pronounced differences in these small RNA levels will succeed to fertilize the eggs and contribute to the phenotypic differences among the pups from control and treated male mice?
Okay, so I'm not sure where that first statement came from, that there appears to be a lot of sperm to sperm variability, but there could well be a lot of sperm to sperm variability. Single cell small RNA cloning protocols are few and far between, and sperm would be very hard to do in them. So we don't have a lot of single sperm epi variability information, but there could be. And so let's just take as granted your hypothesis. Anonymous person.
Yeah.
So that's a question we're quite interested in. And you could also imagine that there are ways that you would miss, if you only had a subset of sperm that would do fertilization, you could miss other, when we say there's no cytosine methylation changes, maybe 1% of sperm do have cytosine methylation changes, and only that 1% of sperm does stuff. So that's an active and interesting area to think about. The best argument against it is that, so there are multiple stages at which sperm are selected. Females choose their mates. After ejaculation, only a couple thousand sperm make it past so soon after mating, the uterotubular junction, so between the uterus and the fallopian tubes, inflames shut. Only 1,000 or so sperm get through that. And then they end up sticking to the epithelium or swimming around in little diverticulae, depending on the mammal. And they get released in waves of 50 until you've fertilized your oocytes. And then, of course, of those 50, maybe only one. So there are lots of places where you could select special sperm. The argument against this kind of model is that we do IVF, so we at least eliminate the selection processes at the UT junction and the release. And some people do paternal effects through ICSI. And so when you do ICSI, you're just picking random sperm and injecting the heads. So, unless you accidentally pick the 1% of sperm that have been epigenetically reprogrammed, you are really eliminating all those selective processes. And because people have gotten their phenotypes to go through ICSI, it argues that sperm are penetrantly reprogrammed. So that's the best evidence against the heterogeneity and special sperm idea. It's a good idea, but there's some evidence against it.
Have you tried like a competitive experiment in the very last experiment you talked about with the turf add back?
We haven't. So we've thought about some of these kinds of things, where you could take a red animal and a green animal and then, mix their embryos or mix their sperm and, in different scenarios. We just haven't done any of those.
Okay. Okay. Nora Kotaja, oh, sorry.
Nora.
Sorry. Would you expect to have different turf profile in sperm after very short, couple of hours versus long days abstinence in human?
Let's see. I don't know that I have a hypothesis about that. I suppose you could imagine that after multiple ejaculates, so at least with rabbits, if you mate them like 50 times, you empty the cotta, but you actually don't empty the corpus or caput, is my understanding. And so what that means is that if you were to sort of completely empty the tanks, then the sperm that will then be refilling the cotta at least will have already been filled up with the caput and corpus small RNA contents. So the only RNAs you'd expect to be affected by mating status would be the sort of very end cotta specific RNAs, which is just 50 to 100 microRNAs in turf Valcac. And we have actually done mating and recovery in measured small RNAs, and we don't see a lot of changes, but we only plug one female. We haven't made a mouse, attempt to plug 10 females and then see small RNA changes. But at least the way we did it, not a big difference in terms of small RNA levels. Could be done more carefully than we did.
Okay. Shui Shen says, hi, Ollie, too bad that you did not give your talk on your treadmill. Great talk in any case. Have you thought of where is the source of the small RNAs loaded to the sperm? Have you looked for them in the brain?
Yeah, so the way we're thinking about it right now is that small RNAs produced in the epididymis are trafficked over to sperm, and anything that's going on in terms of the brain communication to the reproductive tract, the way we think about it is that the brain would ship, whether it's an mRNA or a small RNA or even proteins, those, are clearly received by the epididymal epithelium because we get the Cre recombination in those cells. So we think of whatever the brain ships over as altering epididymal physiology / small RNA production, and then the epididymis makes the RNAs to ship to sperm. There are 2 papers that have claimed that expressing RNAs in the brain lead to those RNAs being detectable in sperm. I'm very skeptical of both of them, the way they measure stuff and the way they claim they don't see contamination. But anyway, there's some people have claimed this. We haven't found any evidence for that kind of idea. And generally speaking, sperm have a very characteristic RNA profile, like they degrade most of their mRNAs and and you don't see lots of neuronal mRNAs in sperm. So I don't think there's a lot of reason to think in mammals that things synthesized in the brain actually end up in sperm. We think if the brain plays a role in the process, it's by modulating the epididymal production. And even the idea that the epididymis produces RNAs to ship to sperm, which again, we've spent a decade working on, I still cannot be 100% convinced it's right. So even that idea is something that if I'm skeptical, you guys should be skeptical. But anyway, so at least as far as the brain goes, we would think it would be the epididymis would be interposed along the way.
Middleman, yeah, okay. Okay, Joanna says, very nice talk. How is selected the information what is worth to be transmitted? That philosophical. Yeah, how is information selected in terms of value?
Right,
What to be transmitted?
right. So that's a really good question. And that sort of boils down to in some sense, it's very similar to the question of what's the bandwidth. So, do you tell your kids about some, integrated quality of life? Maybe just the amount of cortisol you've experienced over the last decade for human, or do you tell your kids about, the levels of nicotine and the levels of cadmium and stuff like that? We think that what you tell your kids is very blunt. And then I guess in that sense, then the next question becomes, okay, but how many things that push down on your quality of life estimate, and how many things push up on your quality of life estimate end up being used by the male reproductive tract? And I would love to know the answer to that question. I don't have any, I have no evidence for an answer at any end of the spectrum.
Seems like to me that many of the studies are focused on metabolic. Do you think that is just that those are the best readouts we have? Or is it a bias?
It's in part because I was giving the talk and I have mostly focused on diet to metabolism. If Tracy Bale or Isabel Mansui or Larry Feig had given the talk, it would have been about stress.
Yeah, mental health. Okay.
Yeah, one thing that I think is worth being aware of is that, those of us who study diet tend to see metabolic effects in kids, or tend to measure those and see those. People who study paternal stress will tend to see, anxiety-related behaviors in kids. But when you look under someone else's streetlight, you'll see their phenotype. So the people who study paternal stress see glucose control when they bother to check. So that is further evidence for the low bandwidth idea that you're maybe just programming a pleiotropic, growth versus stress versus growth axis in the kids.
Right. Right. Okay. Jason Lynn says, if the brain is sensing the environment and transmitting this information to the sperm through an accessory organ, how could this information be sensed in organisms without a brain? Do you think the bandwidth is greater when an organism has a brain?
So that's a good question. I haven't really thought about it because most of the paternal effects literature, it's surprisingly mammals are among the first places for really good paternal effects stimuli. There are lots of good epigenetic inheritance systems. Like if you inject double-stranded RNA into worms, they pass on silencing, RNAi. But that's not a stimulus, that's injecting a worm with RNA. But so for environment-induced intergenerational stuff, mammals have been studied for the longest. But most of the other studies in this domain, there's now, there's a good fly literature, there's a worm literature, other large mammals. So all of these are things that have brains. The only kinds of studies that are done in brainless organisms are in plants. Plants are really good at epigenetic inheritance. Plants have very little evidence for environmental stimuli affecting the next generation. There's a bit of work on drought, intergenerational and transgenerational drought, but outside of a little bit of that stuff, there actually isn't much evidence that plants, program their pollen and eggs in response to whatever photoperiod or drought or something. So I guess that's, that is correlated with the idea that the brain would be required, but I don't really think it's great evidence that for or against the role of the brain in the process.
Okay. All right, we got 3 more. Okay. Siham Chalufi says, why do you think the epididymis is protected from cancer? Does it have stem cells?
Good question. My understanding, and this is a bit outside my field, is that the basal cells, which are the cells outside of the epithelial tube, may be the stem cells for the epididymis. This is work from Daniel St. Cyr, and I don't know it very well. I have no idea why it doesn't get cancer. It's, it's tempting to speculate it has something to do with the fact that it makes huge amounts of tRNA fragments. tRNA fragments are cropping up in cancer systems at some frequency. But really, I just have no earthly idea. If I ever, run into trouble getting funded by NICHD and other people, maybe I'll try to figure out if there's a way to ask the cancer question. But basically, I have no idea.
Okay. Ralph Shoet says, does the innervation of the epididymis influence its instruction of the sperm?
So the innervation in the epididymis is in the cauda, because that's where you get the muscle contraction. So it is the case that most of the recombination with the various Cree drivers is occurring in the innervated part. However, in some of the stereotactic injections we did, we would inject one hemisphere and we would see recombination in both epididymes, as opposed to just the contralateral or the ipsilateral. And the ability to transfer the information, as inefficient as it has been, still also argues against the Cree being dropped, immediately at neuromuscular junctions or something. So we don't think the communication is through innervation. It's also the case that the prefrontal cortex, it's not spinal cord neurons that are getting the Cree, it's the brain itself.
Mm-hmm.
So well, all of those things kind of argue against the idea that the Cree transfer is by what do you call it? The neurotransmitter vesicles.
Right. Okay. Mark Wasidlo says, opening the Pandora box, what about changes in tRNA modifications transmitted by those exosomes?
Yeah. So one of the things Upasna started her lab to do was to ask the question of how the levels of tRNA fragments are regulated by environment. And we know that some nucleotide modifications on tRNAs affect cleavage by the various nucleases, DNMT2 being the really famous one. First of all, nucleotide modifications could affect just the cleavage of specific entities. There's also work, this guy Chi Chen, who's probably not far from you guys. I'm not sure how far Riverside is from San Diego. He is really committed to the idea that actually it's not the levels of tRNA fragments, but it's their modifications that are transmitted. I'm a bit skeptical, but not completely skeptical of that. And one thing that I wonder about is the extent to which some of the nucleotide modifications he's interested in don't necessarily affect the function of the tRNA fragments, but maybe just affect their stability. So in the end, they affect the levels. And he's also put out that idea. So let's see. What was the other thing I was going to say about that? Yeah, I lost it. Anyway, it's a good idea. Oh, I will say, so we had with our favorite tRNA fragment in some of our assays, we made the tRNA fragment synthetically without nucleotide modifications or with the correct ones. For one or two assays, the modified tRNA fragment was more efficient at a given dose than the unmodified. But and at first I was like really keen on that idea. But the more things we measured with it, the more they acted the same. So if anything, it would be a sort of like mild change in, stability or efficacy or whatever.
Right.
So at least in our hands, it's not a big thing, but it's an idea worth exploring.
Okay, great. Okay, that brings us to the end of the questions. And yeah, you thought this was not going to be interesting to our crowd, but turns out I think this is the longest Q&A we've had. So, so inherently interesting. Yes. Thank you so much. This was really fun.
Well,
and
thank you for inviting me. Thank you all of you who stayed to the bitter end. I wish I could see you all in little boxes, but anyway, thanks so much, Diana.
Yeah, we'll have you out in person when this all normalizes.